Good morning. This is Michael Dong on video episode number 26 on polymer analysis by HPLC and also GPC, standing for gel permeation chromatography. This is my agenda. Three different sections. First is overview, mentioning also about chemical industry in addition to the polymer industry. Second is a fundamental summary of GPC, gel permeation chromatography, talking on fundamentals, molecular weight averages, the calibration process, something called MWD, standing for molecular weight distribution, and several applications. Finally, a few common operation and topics such as the GPC operation, mobile phase selection, and a couple of case study on two-dimensional liquid chromatography, 2DLC, and reverse phase chromatography of polymer additives. Here's a brief overview of the chemical and the plastic industry where HPLC is widely used. I would comment the fact that for chemical industry, HPLC, there's some similarity with those of testing in pharmaceutical, including things such as the potency assay for the main components, purity testing for synthetic chemicals, and also testing of raw material, precursors, monomers, surfactants, detergents, and dyes. In the plastic or polymer industry, using organic polymer, a huge application of GPC for polymer characterization during both product research and quality control. Reverse phase can be used for determination of additives and characterization of lower molecular weight polymer and also oligomers. Just a comment on a niche and also a very important subject on leachable and extractable analysis. These are indispensable assays for food and pharmaceutical packaging applications to protect the public health, mostly by HPLC in couple with high resolution mass spec. However, we will not cover these aspects in this video. Some overview and terminology. GPC occasionally also being the terminology is synonymous with size exclusion chromatography, size exclusion. GPC specifically when it's used for polymer chemists and plastic engineers, we call them GPC. Separation is by effective molecular size or hydrodynamic volume. So even though that we like to equate them with molecular weight, the actual correlation with the hydrodynamic volume or the effective size in solution. So this distinction. So you have to have a good set of calibration standard, which we will talk about that later. Typically in GPC, we use a control pour particle size material. Typically, these are cross-linked gels, so resins, using copolymer polystyrene with divinyl benzenes. Also for aqueous GPC using some different materials. GPC system is a very simple HPL system. It needs to have a very precise pump to give you accurate molecular weight calibration. Typical mobile phase THF, tetrahydrofuran, or toluene, and generally a refractive index detector is being used. And some comment on GPC using for large macromolecules, and occasionally we can use it for smaller molecular weight for purification or sample preparation. It's very important in GPC to in the design, in the method development portion of it, to eliminate any absorptive interaction of the solute with the support. Otherwise, you will not get a true molecular weight correlation. So it's very important to eliminate those any active site interaction using mobile phase additives. Further comments on GPC on molecular weight averages. So we mentioned molecular weight averages and molecular weight distributions to give you a profiling of the sample. And there's many different type of averages, which will be defined later. And in order to calculate these molecular weight averages, one needs a specialized GPC software, which typically a additional optional software package linked to the CDS to chromatography data system. So in general, for a large range characterization, one uses something called a mixed bed column, because most of the GPC are packed with controlled pore size, and typically you would use very small pore size for smaller weight polymer. However, for a longer range for screening purposes, a mixed bed column with different pore size packing material is being used. We show an example later on. In order to get a good molecular weight characterization, one need to calibrate the column set with a set of calibration standard, which is appropriate for the sample size. Unfortunately, some of these standards are not readily available, with the exception of polystyrenes, which are a very standard set of calibration standard. In those cases, one may use a universal calibration or other techniques such as a multiple light scattering detector or mass spectrometry. Some of the case study on universal calibration will be mentioned later. Some characteristic of GPC. We mentioned that GPC is best used for high molecular weight components, say molecular weight above 1,000, and probably the range is 1,000 to 10 million molecular weight. In GPC, all solute should elute before the solvent front, where's the total permeation volume. And there should be no or very low absorption interaction between the support and the solute. A GPC is typically what we call a moderate resolution because only can happen under isocratic condition, and the capacity is rather limited. Therefore, it's very difficult to separate size differences less than 10%. And we mentioned that the column set or the column must be first calibrating using a narrowed range molecular weight standard. I'll give you some examples later on. Uses for sample fractionations, in addition to polymer characterization, sample profiling for different plastics, and characterization of molecular weight averages, and also distribution of polymers. This slide was shown earlier in the fundamental in the second video of this program, demonstrating the retention mechanism of GPC by molecular size sieving kind of process. So in GPC, we have a controlled pore size support with hopefully no interaction between the solute. Now in this support, the large polymer cannot penetrate the size, therefore it was eluted later. Therefore it's eluted earlier because it cannot go through, penetrate all the pore. while the smaller molecular weight solute would go into all the pores and elute it um, later. So this is the mechanism called size exclusion chromatography. So in size exclusion, the mechanism is by size exclusion, separates by molecular size rather than molecular weight. A large molecule cannot enter the smaller pore and therefore elute it first. So here is an application. In this case, it's used for screening purposes. We use a polymer laboratory, which is one of the first developer of very high quality GPC columns. This company is located in the UK, and GPC columns are typically packed with cross-linked polystyrene and diventyl benzene copolymers. A Polymer Lab from UK was acquired later on by Varian in 2005 and later on by Agilent, which acquired Varian in 2010. So here is the separation of different polystyrene standard from 1.6 million, which is peak number 1. This is the large molecular weight polystyrene, to number 9, which is the oligomer, monodispersed oligomer with a molecular weight of 580. You can see that the smaller molecular weight polystyrene eluted later. Actually, the diluent orthochlorobenzene is the latest elution. The detection is, in this case, is a UV detector, detection at 265. And the mobile phase used is THF. Shown here is a set of calibration standard using Polymer Lab Gel's GPC column. One can see that there are a large variety of a column with different pore size, from 1 million angstrom pore size to 50 angstrom pore size. So typically for smaller molecular weight polymer, one would use smaller pore size. For screening, one would use a mixed bed GPC column. Some of these examples shown here is published in this article written by Wilhard Reuter and myself in 1991. Some case study on polystyrene oligomer using a small pore, 100 angstrom pore size, showing a separation of some of the smaller molecular weight polystyrene. This is an example case study of polyvinyl chloride, both for identification and isolation of plasticize. PVC use require quite a different type of plasticize shown here, and here is a way to isolate the excluded polymer shown here, and also one get a GPC separation of the different type of phthalates used commonly for PVC. A different example shown here on the separation of epoxy resin, which is derived from two polymer, epichlorohydrin and also bisphenol A. Is, when you make an epoxy, you use these two material, mix them together and form a very strong epoxy. And here is the operating conditions using a 500 angstrom two PL gel coupled together using refractive depth detector. This is separation of shell apical, which is a very common epoxy resin. In this case study, we compare the resolution of bisphenol A epoxy using a conventional GPC, using larger particle column. In 2016, Waters Associate Waters Corporation introduced a system called APC, standing for Acuity Advanced Polymer Chromatography, using a smaller sub 3 micron particle column, GPC column, coupled together with lower dispersion systems, using a high performance refractive index detector, and demonstrate the ability to show a much faster separation, shown here in about 6 minutes, on the same epoxy resin sample, giving you a higher resolution and faster analysis. Switching gear towards aqueous GPC. For aqueous soluble polymer, water soluble polymer, such as classification here, polyvinyl chloride, PEG, polyacrylates, and the polysaccharides. Typically, one uses a set of column packed with cross-linked polyhydroxy methacrylate or polymethacrylates to get more hydrophilicity in the column materials. So use different set of column, obviously, and use a aqueous buffer in the mobile phase. Shown here is an assay of aqueous GPC of polyvinyl chloride using this set of aquagel column from Polymer Laboratories. Some general comment on characterization of molecular weight distribution to monitor polymer changes during the processing of polymerization, processing, and possible degradation. Shown here is a case study of GPC analysis of alkyl resin. Alkyl resins are adhesive. And shown here, even without calibration, if one get a molecular weight distribution profile, one can often distinguish different, in terms of the quality of the adhesive. Here shown that a good batch is a batch with less high molecular material, meaning that there is a better adhesive property, while the bad batch typically have worse quality due to the fact that very high molecular weight material in there. Shown here is the a GPC analysis of a polystyrene foam sample. Some of you might be familiar with these coffee cups, disposable coffee cup using polystyrene. These are made from a polystyrene foam. And this is using a mixed bed 5 micron gel column. These are the conditions. And using a GPC software program, one can get a different type of molecular weight averages shown here, plus a very important parameter called the polydispersity to measure how narrow the distribution or how wide is the distribution of the molecular weight distribution. Here are the actual calculations being shown here. Some are called the number averages, weight averages, Z or the in terms of the polymer length averages, viscosity averages, and polydispersible indexes shown here. In this table, which is derived from this book chapter by Yao et al. In 1979, demonstrating some of the effect of molecular weight and molecular weight distribution on polymer property in terms of properties such as tensile strength, toughness, hardness, melt viscosity, adhesion, and things like that, solubility, shown here. So these are important for plastic processing. We mentioned that for GPC calibration, it is important to use the right set of calibration standard. Now, unfortunately, with the exception of polystyrene, some of these sets are not readily available. In those cases, one have to use a calibration situation called the universal calibration, shown here in this particular reference over here. So attendees refer to this particular references for detail on how to carry out the universal calibration. So one used a equation called the Mark-Hewing equation, which is relating something called intrinsic viscosity of the polymer with molecular weight, and it required the accurate determination of two Mark-Hewing constant, the K and alpha here, to allow you to get these calibration standards. Some comments on GPC operation on mobile phase characteristic. So, so the column material must there's some requirement. The system component cannot be corroded by the mobile phases. Detector typically UV or refractive index. Sample must be able to dissolve the entire polymer sample without degradation. So some of the desirable characteristic, and also typically the most common mobile phase is THF, tetrahydrofuran. These are typically stabilized with the addition of BHT. A common GPC mobile phase solvent are shown here in order of increasing polarity. So from hexane, we mentioned about toluene and THF being very common, and some of the other ones. I mentioned that for polyolefin or polyethylene and polypropylene, one required to use something called the high temperature GPC. Very few manufacturers would manufacture that. One of them that I know of is GPC from Waters Corporation. So shown here are the mobile phase for very high temperature GPC at 140 using orthodichlorobenzene, which is a very nasty and toxic solvent on that. I mentioned some pretreatment advice for GPC. So typically one have to sample, you dry, mix, grind representative polymer sample. You dissolve the sample in appropriate solvent by agitation, typically stirring or magnetic stirrer. One should try to dissolve the sample in mobile phase if possible. Make sure you examine the sample solution, make sure you have complete the solution. Filter the samples through some membrane filters. Very important not to ultrasonicate because polymer chain might be fractured during that process. A couple more case study or application. Shown here is the HPLC analysis of polyethylene additive shown here as the component. So polyolefin require quite a bit of different additives. Some is used for antioxidants, others are mold releasing agent. So shown here is a gradient analysis using reverse phase. And the extraction of the additive is by a saucer type extraction using cyclohexane. Finally, I'd like to show a very complex polyether polyol with two-dimensional LC coming from this particular references. So this is an area of very active research using two-dimensional LC in conjunction with mass spectrometry, hyphenated with high resolution mass spectrometry, to characterize some of these polymer materials to give you very high resolution. Final application is surfactant analysis. I want to mention that this particular column is very specialized, very niche column. column is manufactured by Thermo Scientific, called the acclaimed surfactant column. And shown here is different type of surfactant material using something called a mixed mode chromatography. Okay, the retention is by a mixed mode of hydrophobic interaction, size exclusion, and ionic interaction type of chromatography. Here is my list of references. Some of these are highlighted in red are the one which is more important. And here is the end of episode 26 on polymer analysis by HPLC and also as well as GPC, both fundamental and applications. On that note, please subscribe to the channel for future videos, and we welcome LCGC as a channel collaborator. I'll see you next time.